Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 1.4, carbohydrates. Objectives for this lesson are to describe the structure and function of carbohydrates, especially discussing the monomers that form carbohydrates and how they do so. And the suggested skill is 1A, concept explanation, describe biological concepts and or processes. So, monomers and polymers of carbohydrates. And carbohydrates consist of CHO, as we talked about in 1.3, meaning they're made up of carbon, hydrogen, and oxygen atoms. So, a carbohydrate is also known as a sugar or a saccharide. And so, the monomer is a monosaccharide or single sugar, while the polymer is a polysaccharide or many sugar. And monosaccharides are either a one ring or a one carbon chain structure. So for instance, looking at the glucose molecule over here, we are seeing a carbon ring, and each one of these points on the ring is going to be a carbon atom. Whereas oftentimes there could be a carbon chain, where again each point of the chain will be a carbon atom. And monosaccharides include glucose, galactose, and fructose. As you can see, a lot of the sugars are going to end in ose. And we have disaccharides, which are two sugar, such as lactose and maltose. And carbohydrates are connected together through covalent bonds called glycosidic linkages, such as this bond here between this galactose and glucose molecule through this oxygen molecule. Oxygen atom. And as you can see, this is going to be an example of dehydration synthesis, which allowed the connection of these two monosaccharides to form a disaccharide. It's important to remember the chemical formula for glucose. That's one of the most common carbohydrates, and that's going to be C6H12O6. And this also shows us a kind of a trick in the chemical makeup of carbohydrates, which is they often have a 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen. So for every one carbon molecule, there's going to be one hydrogen molecule and one oxygen molecule within the carbohydrate. Now that we've covered the basic monomers of carbohydrates, let's discuss some of their properties. So carbohydrates can also have isomers. And as you recall, or maybe not recall from 1.0, isomers have the same chemical formula, but different structures. So we have structural isomers, where there's a difference in the arrangement of the bonds, and enantiomers, which are mirror images. So if you look up here at the top right, we have a glucose molecule, and then we have a galactose molecule, which is almost it's just a restructuring of the bonds, and same thing with the fructose molecule. They all have the same chemical composition, but the hydroxyl group is on the left here. Over here there's a hydroxyl group on the right, but we also have this carbon double bound to an oxygen. And down here we have an enantiomer. It's just a mirror image. So we have these isomers that have the same chemical formula, but even though they have a same formula, they have a different structure, which will then contribute to a different function. Now, carbohydrates can also have different kinds of linkages. And what I mean by linkages are where two monomers will link together through dehydration synthesis. So this glycosidic linkage or glycosidic bond which connects two monomers can either be on the 1 and 4 carbons. If we're looking in the carbon chain starting from the oxygen molecule, this is our first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon right up here. So there can either be a 1,4 linkage or a 1,6 linkage, which again is going to change the structure of the overall carbohydrate molecule just a little bit, even if all the monomers are the same. Within having a similar structure or even a similar chemical formula, we can have the exact same monomer, but result in a different polymer when we talk about alpha versus beta glucose. So there are two ring structures for glucose, one where the hydroxyl group is attached to the one carbon and is either positioned up or down. So over here, within starch, we have 1,4 linkages of alpha glucose monomers. And in this case, the hydroxyl group is always going to be facing down. And then in cellulose, we have the exact same makeup of glucose molecules, and they're not structurally any different the glucose molecules. However, there's going to be a different linkage where the hydroxyl groups are going to be positioned up every other glucose molecule. So as you can see, that's going to change the bonding because the bonding is going to switch every ring. And this is a difference between alpha and beta glucose, where we have the same chemical formula and structure of the monomer, resulting in a different overall polymer, just by flipping the monosaccharide. Now, also extremely important are the function of carbohydrates. So we talked about the different structures that a carbohydrate can create, but now let's link this structure to function. So carbohydrates do two things. They first of all store short-term energy, and two, help create support structure. So energy is stored in plants as starch and in animals as glycogen, and these are both polymers of glucose. And glucose is our basic unit of energy, or basic unit of sugar, and that is going to be then stored within either starch or glycogen, depending on whether you have a plant or animal. Now cellulose, another polymer of glucose, is a major component in plant cell walls, which provides structural support to the plant cells. And chitin makes up exoskeletons of lobsters, insects, and fungi. So carbohydrates do these two things, store energy and support structure. Now why do I say short-term energy? Well, this is because lipids store long-term energy, and we'll talk about why in the lipid video. But now let's look at how the different functions of carbohydrates are linked to the different structures of carbohydrate polymers. So here we have three different kinds of carbohydrates. We have cellulose, we have starch, and we have glycogen. And remember, we discussed that starch and glycogen are our energy storing molecules, while cellulose is within plant walls. And so here's our cellulose pictures right here. And as you can see, we have a sheet here. It's going to be a very strong compound because we have hydrogen bonds attaching these rows of cellulose that are all covalently bonded. These are called microfibrils, and we have these molecular chains that are all in a very rigid structure. And again, because cellulose is made to support the structure of plants in the plant cell wall, this makes sense that it would be such a rigid and structural organization within the molecule. Whereas starch and glycogen, as you can see here and here, we have a lot of branches rather than rings. And through these lines and branches, we can see that starch is all in one row, so bonds can easily be cleaved, releasing the energy. And when we want to get this energy back from the starch or glycogen that we've stored, this structure allows for easy cleavage, which allows the release of energy from these bonds. Same thing for glycogen. All of these branches are made so that they can be easily snapped off. You can easily break the bonds between them because they're just branches. There's no interlocking structure. And so this brings us to the triple AAA rule, appearance affects activity. And this is also called structure affects function or form follows function, and it just means that the structure of a macromolecule or of a compound is going to then affect its function. And we can see this tie here comparing cellulose, starch, and glycogen. So, carbohydrates review. If you didn't get all the fine points of carbohydrate structure, such as isomers and the alpha and beta glucose, that's okay. Main idea, monosaccharides make up, which are the monomers of carbohydrates, make up carbohydrates, which are a polymer, and these carbohydrates have a function of storing short-term energy and providing structural support. And of course, you got to know your AAA rule, appearance affects activity. We'll see you in 1.5 to talk about lipids.